So I'm so much, excited to say give little information about Dr. Shu that I have come across. So though she is very young, I'm seeing that she had achieved so many as a research scholar and even during her education also. And her research interests are mathematical biology and numerical analysis and scientific computing. As I said, Dr. Chu, we have a lot of people working in applied field in the department. I'm really very, very happy to see you. And I'm looking forward a day you come and visit us. But we will postpone little towards students, I would like to say. So she has so many publications, talks, she is even as a reviewer board for several journals also, so which is very impressive. Students, if you would like to see, go through and see her CV, Google Scholar, everything is available. So it's very, very interesting. I'm very happy. Thank you for being here. So now, yes, please take over the floor. Yeah, thank you.
Thanks, Dr. Panakala, for the nice introduction, and thank you all for being here. I plan to make this talk like a bit shorter than a regular 7-minute talk, so feel free to interrupt me like for questions, etc. So, okay, so let me start the slides. Okay, so first of all, I wanted to acknowledge my collaborators on this project. So this has really been like a long project, and what I'm going to present is only like the most recent portion of the project. So we all know that mosquitoes cause a lot of human suffering, and the specific mosquito species that we focus on for this talk is the Aedes aegypti mosquito, which can transmit dengue fever, chikungunya, or Zika. And there are many mosquito controls that people have come up with so far. So we can use the insect spraying to kill the mosquitoes. We can remove their breeding habitats, such as like scrape tires or water containers. Those are the place, like the homes for the mosquitoes. There's also biocontrol, such as the sterile insect technique, SIT, that I listed here. So those strategies, they are aimed to kill the mosquitoes, right? Those are more the population suppression strategies. And well, unfortunately, like typically these population suppression strategies require long-term mitigation program to maintain their effectiveness over the time. That is, once you stop doing like insecticide spraying, then there's a chance that mosquito may come back, and then they may just grow back to the previous abundance level, which is not ideal for disease control. So for the Wolbachia strategy that we focus on for this talk, it is using a slightly different mindset. So Wolbachia is a bacteria itself that's been found in many insect species, including some mosquito ones. And the point is, like, when the mosquitoes are carrying this Wolbachia bacteria, they're less efficient in transmitting, say, dengue virus to humans. So let me just do a brief walk you through like how does that work. So for example, this is a transmission cycle of mosquito-borne disease. You see a mosquito, it bites a sick person and carry the infection and then pass it to a healthy person, make them sick. So now when the mosquito is carrying this Wolbachia, so this is a Wolbachia carrying mosquitoes, and then it bites a sick person at this stage, and then it bites another healthy person, it's no longer able to transmit the virus to the healthy person. So in another word, for this strategy, we are not killing mosquitoes. We are infecting them with this bacteria so that they are no longer the vector for this transmission cycle. Well, ideally, we wanted to make all the mosquitoes carrying these Wolbachia bacteria so that they are useless for disease transmission. Well, the thing is, the Wolbachia infection is not naturally found in the wild Aedes aegypti mosquitoes. So what people do is they need to rear those like mosquitoes in the lab and release them to the field. And when the Wolbachia carrying mosquitoes are mate with the wild type field mosquitoes, the Wolbachia infection can be passed from infected female mosquito to their offspring. So this is a maternal transmission route for this infection. And the goal will be eventually all the mosquitoes are carrying this Wolbachia infection. So we're going to do population replacement strategy. And if the infection can be established, then the infection can be like passed from generation to generation without much effort from human side. So from this perspective, this is a promising strategy to be more sustainable compared to the traditional ones. And in fact, many field trials has been implemented around the world for control the dengue epidemics. And people do report promising results from these trials. So when it comes to field releases, then a key question that we need to address is how many of these mosquitoes we need to release, right? So if we just release one mosquito, it's not going to save the world. So we know that it is difficult to create this Wolbachia infection in the field because the infected mosquitoes, they're less fit compared to the natural ones. That is typically that happens in the infected female mosquito. They live a shorter lifespan and they produce fewer offspring. So if we just introduce a couple of mosquitoes, then they're not as fit or competitive as the natural mosquitoes, so they will be wiped out very soon. Biological cage experiments has shown that there's a threshold level. So there's like a minimum amount of mosquitoes you need to release in order to have a sustainable infection. We will see also in a second that the mathematical model also confirmed that there is a threshold condition, which is realized as this fraction of infection. So when the infection rate is above some level, infection can establish and spread out. And when the infection is below a threshold, then the infection just collapse and die out eventually. Another significant feature we want to include when we talk about field release is that mosquito, they fly around. So the spatial dynamics can be important. So that coming from the flying dynamics, the mosquito dispersion, right? That can lead to the spatial heterogeneity in terms of the mixing. So you may have lots of infection near the release center, but not much when it's far away from the release center. There's also spatial heterogeneity coming from the environmental setting. You know, there is a lake there, so you have more water and more mosquitoes, but you may not have that elsewhere. So that can also impact how we design the field release. So our goal is to figure out the threshold when we have this spatial dynamics as well. So I will first look at the Wolbachia transmission dynamics in mosquitoes. So here is a diagram. We have the female mosquito, we have male mosquito, and when they mate, they produce offspring. So the color indicate the infection status of the mosquitoes. So if they're all wild type, like they're Wolbachia free, then there's no infection. When the male mosquito is carrying Wolbachia, but the female is natural, then the offspring they reproduced will not be viable. So there will be offspring, but they cannot be like developed. So this is induced by the cytoplasmic incompatibility from the Wolbachia infection. In the next two cases, when the female is carrying Wolbachia, regardless of the male infection status, there's a high fraction of the offspring that will carry this Wolbachia infection. So this infection, the rate of infection is referred as maternal transmission rate. Based on this transmission diagram, we proposed this compartmental model. So we divide the whole mosquito population into different groups, right? We have the male mosquito, female, they produce eggs in the water, and egg mosquito can become larvae, pupae, and then it can emerge from the water and become flying male or female mosquito. We have the top row is the Wolbachia uninfected ones, so they're natural mosquitoes. We have the Wolbachia infected ones in the bottom row. So for example, depending on the mating combination, right? So if I have the infected females here, then regardless of the male status, the female can produce like a high fraction of infected offspring, or there's a chance that the female can produce like the leakage there with some uninfected offspring. So this model here captures some key Wolbachia features such as the maternal transmission rate and also the cytoplasm incompatibility. We also incorporate the fitness cost, which we mentioned earlier. That is an important impact on the female mosquitoes. So we are going to have different death rates here for the Wolbachia-infected cohort, so that to incorporate this reduced lifespan and reduced reproduction. Okay. So let's get to the math part. So here is my equation. So the birth rate of the uninfected eggs that can only come in from uninfected male mating with uninfected female. So if I have uninfected female with a probability that it mate with an uninfected male, and then produce eggs at this egg laying rate VU, and then those eggs can leave the compartment at this hatching rate here, or they can die. Similarly, for the infected cohort, the infected eggs can only be produced when the female mosquitoes here mating with the male mosquito. So that's the female mosquito producing an egg at the VW rate, can hatch, can die. When there is imperfect maternal transmission, meaning that the infected female can produce both the infected and the leakage of uninfected, then we're going to modify this by this 1 minus VW. So VW is the maternal transmission rate, say 90%. So there's a leakage here, that is 10% goes here, and then 90% goes here. So that's the VW in the front. Then we derive the equations for the larvae stages and the adult stages. So this is pretty typical. So you have the inflow minus the outflow. I want to point out that we do incorporate some density dependence on the larvae stage here using this logistic growth term. So this is because biologically people observe that there's lots of larvae competition going on at this stage where larvae are competing with each other for resources. So for the relevancy of biology, we incorporate that term here, which also regularizes the system. So doing the onto analysis of this ODE model, so we first calculate the basic reproduction number, R0. So if you're working with like epidemiology, or if you learn from the news on the COVID, right, R0 is a quantity that will give you an idea whether the disease will spread out or not. So here you can consider Wolbachia is a disease for mosquitoes. So we're infecting mosquitoes with this pathogen, and for one Wolbachia-infected mosquito, so R0 describes how many additional infection that it can create. So using a standard like next generation matrix approach, we derive analytically the expression for R0, and we're plugging some biologically baseline value for the parameters. We see that actually the R0 is less than 1, it's around 0.7. So initially we're saying, okay, does that suggest that there's no chance for the Wolbachia to be spreading among the mosquitoes to be useful for disease control? But actually, this is not true, because the Wolbachia only describes the initial phase of the disease transmission. So later on, we do some bifurcation analysis to check the dynamics near other steady states of the system. So here I show you this diagram. On the x-axis is the R0. On the y-axis is the Wolbachia infection prevalence in female mosquito. The system has 3 steady states, biologically relevant steady states. So there's Wolbachia-free, 0 infection steady states, which is stable. There is also a high Wolbachia prevalence stable endemic state. There's an unstable endemic state connecting these 2 states. So if we are able to bring the Wolbachia prevalence above this unstable state, then the infection actually can grow and approach to the stable coverage. If we only introduce a small amount of Wolbachia infection, so that we are just below this state, then it will collapse. So the Wolbachia will disappear. So in another word, this unstable steady state is a threshold Wolbachia coverage that we need to have in order for the infection to spread. At the baseline scenario, R0 is around 0.7. So this gives us the threshold coverage with a value of 25%. And then now we need to incorporate the spatial dynamics. Okay. So I mentioned at the beginning that the cage experiments from the biology, that is, you assume the infected and uninfected mosquito, they just homogeneously mixed in a cage. So in that scenario, the 8 ODE model that we just described can be suitable for describing the dynamics. So we see the bifurcation plot, which is saying that all the mosquitoes, like essentially this is saying there's no spatial dynamics. And we see there's 25% of a threshold. But then when it comes to field releases, it's no longer valid. So I will just show you a set of simulation here to see like the impact of spatial dynamics. So I take my ODE model and just attach to a simple diffusion term here, make it like a reaction diffusion system. On the left, I'm going to simulate a field release of step shape. So the X dimension is the spatial field release dimension, and the Y-axis is the fraction of infection. So the red dashed line here is the 25% threshold we identified. So I see that, okay, I release above that threshold 25%, and the infection is established towards the top and then propagating in the nearby region. So that's a successful establishment. Now I change the configuration by releasing a lot more at the center, but in a much narrower region. And in this case, the infection collapsed. So you see that the OD threshold, or the 25% threshold value that we identified earlier, is no longer like a good indicator saying that if my infection can be established or not. So the threshold, though, it will depend on the spatial dynamics. And also, to make it more realistic, more biologically relevant, we're going to consider the spatial dynamics in 2D, meaning that my mosquito does not just fly to the left or right, they will actually fly in all possible directions in the field. That's like the word we live in, right? Okay, so I'm going to do some modification on the previous 8 ODE model by adding the diffusion terms on these females and males equations. So for these adult stage mosquitoes, they fly around, that leads to this dispersion. Where we don't have any diffusion terms on the aquatic stage, meaning that mosquitoes, they live in the water, so they don't really move around that much. So we assume no diffusion there. And everything else stays the same. But now when we have PDE models, the analysis become much more complex. So our goal is to try to understand the threshold dynamics. So what amount of infection is needed to sustain and spread this infection? In particular, we're interested in local release. So people are carrying their box of infected mosquitoes and release in a field. So that corresponds to a compact support for the initial condition of the PDE model. For threshold of this PD scenario, okay, this is what we mean. So if I do a field release, if I say it's below threshold, if I release and simulate the infection collapse. Above threshold, meaning that I do the release and then the infection grow and then propagate. Okay. So the threshold, by observing this dynamic, we can actually numerically identify that threshold by just iterating the release level of these these steps, so that when we are at near the threshold release level, then the infection can maintain some shape, not collapse or not grow. These can be better observed if we track the infection level at the peak or at the release center of these curves. So if it's above threshold, then the infection will grow. If we below the threshold, the infection will collapse. So we have this bistable behavior of the system. Additionally, because we have now the spatial dynamics or the spatial dimension in the system, so we actually no longer restricted by a step release, right? We can have steps of different widths, or we can just do a triangle release, or we can carefully align my mosquito so that it is in this elliptical shape of the distribution, which is totally not realistic. But regardless of the shape, we can identify the corresponding threshold release level. And we see that all these threshold levels, when they evolve, they always evolve to this bubble-shaped profile in time. This is not that surprising on the hindsight because we're dealing with a reaction diffusion system. So the reaction part or the OD part of the model, it represents the net growth in the mosquito population. So this will help, like this will push the infection up locally. The diffusion part of the system helps to spread out the infection to a nearby region. So this actually will lower the infection level at each given state, each given spatial location. So the actual bubble shape is a delicate balance between these competing forces. So you have something pushed up, something pushed down, then the threshold is when like these two forces balance each other. So for that sense, we say, okay, instead of doing like a threshold for different spatial profiles, let's just stick with this bubble shapes that is unique across different distribution. And we call this our critical threshold bubble. Now we have the PD threshold and compare that with the previous threshold level from the OD analysis. So this is the PD bubble, which is half of the domain. Because it's symmetric, so I only show you half of that. And this is the 25% from the OD. So we see that when considered spatial dynamic, it requires a lot more infection nearby the release center at 0 here. And you don't need that much of infection when you are far away from that release center, right? And the reason, on the hindsight, again, it's kind of understandable because initially you need a lot more infection near the release center so that you can support the growth of the local establishment of the infection, right? So, and then once established, then it will just propagate outward. So you don't need that much far away from the release center. So we see that this level is much higher than ODE. The ODE threshold is definitely like not an ideal metric to tell whether your infection is successful or not. Then we generalize this discussion into the two-dimensional field. So we have this is our 2D bubble. We can take the cross-section, a slice of that, as this red curve here, and then compare with the 1D PD threshold, right? We see that the 2D bubble at the release center, it is much higher than the 1D diffusion. This is because when we have two-dimensional diffusion, under the same diffusion coefficient, the 2D dimension, like the 2D diffusion is actually much larger than the 1D diffusion because you have a lot more direction to diffuse. So to balance that higher diffusion, then you need a higher concentration of infection initially. So that explains why the 2D threshold is much higher than the 1D. We are also interested in seeing, okay, now we get the threshold. So how does that threshold depends on different model parameters? So that's why we do the sensitivity analysis here. So we consider different metric of the bubble. So we consider the peak of the bubble, the width of the bubble, like how wide the bubble spread out, and also the area under the bubble, which give us an idea like how many infected mosquitoes we need to release. So we consider the peak of the bubble. That give us this PRCC plot here. So PRCC is the technique we used for sensitivity analysis. So the direction, like negative means it's negative correlation, positive is positively correlated, and the magnitude give us like how strong that correlation is. So we see that the red bar, which is the maternal transmission rate, is the most impactful in determining the level of this threshold. Second to that is these parameters with these yellow bars here. So this set of parameters is corresponding to the survivalship of the eggs and then the reproduction of the females. So it is really the reproduction that dynamic that determines the threshold level. When we consider the width of the bubble here, the maternal transmission rate stands out. In fact, maternal transmission stands out for all the quantities that we have considered. And second to that, it is these blue bars here, which represents the diffusion coefficient and the fitness of the males. So overall, we see that the height of the threshold depends on the reproduction and the survivalship of the offspring, whereas the width of the bubble depends more on the mating process from the males and also the diffusion process coming from the flying behavior. We also considered like when the infection is established. Okay, so how fast the infection wave can propagate to the nearby regions. So we consider the wave velocity here, and we also consider the shape of the wave front, that is the width, we say the width of the wave. So again, the maternal transmission rate stands out. And then for the wave velocity, it depends on this yellow category of parameters, which represents the survivalship and the reproduction of the offspring. Whereas the width, the shape of the wave front depends on the diffusion coefficients. Okay. So in the last set of simulations, we're going to simulate a bunch of releasing scenarios with the goal to shed some light on how we can better efficiently design the field release. So often the time, people combine the Wolbachia release with some traditional strategies before the release. For example, here I do the pre-release mitigation on the adults. So first, kill a bunch of the natural adult mosquitoes using the residual spray. And then I can introduce the Wolbachia mosquitoes. So I consider different radius of release. So that's the region that I release my mosquitoes. And I also considered different mitigation release. So this is an area that I'm going to kill the adult mosquitoes. So I vary these two different coverages and see if there's any like particular combination that give us the best, if most efficient release strategy. So this generates this heat map here on the left. So the x-axis is when I vary the mitigation radius. Y-axis is when I vary the release radius. So if you take a vertical slice of this result, okay, that is, I do the mitigation with a fixed area, but I vary how wide of a coverage that I do my Wolbachia release. So these give us the plot in the center. So I see that when I increase the release radius here, by the way, the color here shown on the heat map, it represents the threshold of the Wolbachia release. So that's give us like an idea how efficient the release strategy is. So I see that it decreases around radius 200. So I need small amount of Wolbachia release here, but beyond that, it goes back. So this is like an optimal release radius. When I look at the horizontal slice of this plot, I see that for a fixed like release radius of my Wolbachia, and when I increase the area of killing the natural mosquitoes before the release, it always helps to reduce the number of mosquitoes needed, which is expected. The more effort you put in the mitigation program, the better outcome you get. But the interesting thing is the trend here. So initially, if you don't do any mitigation, then near that region, the reduction is actually pretty slow until you hit a sweet spot here. You see this inflection point here where the release number decreased the fastest, and then beyond that the infection like the threshold actually saturates here. So what it tells for the field people is that when they design the field release, you better hit that sweet spot here, right, to make the most efficient use of your mitigation program. We can also do pre-release mitigation on the aquatic stage. So that is, you can first do the larviciding, right? So that's the corresponding heat map here. We don't see much variation horizontally, meaning that no matter how much of mitigation I do on the larvae initially, it does not impact the Wolbachia threshold by that much. I further do the mitigation on both the larvae and egg stage. I don't see that much impact horizontally. And compared with the previous pre-release mitigation on the adults, I see that targeting the adults before my release is much more impactful. So this is saying that if you want to do pre-release mitigation, you'd better just target adults rather than wasting your resources on the aquatic stages. And on the hindsight, we think that this is also making sense because the goal is to do population replacement. So we want all the mosquitoes be replaced by this Wolbachia cohort. And this population replacement works most efficiently in the adult stages, because at the adult stage, so this is diagram I showed you at the beginning. So at the adult stage, this is my uninfected cohort. This is my infected cohort females. So with the Wolbachia introduced, it's actually killing lots of the natural uninfected offspring. And it actually spread out the infection among the infected offspring. So the replacement is a competition between these two cohorts. So the Wolbachia infection actually facilitate that replacement when we have the adult mating process. So targeting the adults before the release help facilitate that spreading. Then I'm going to show you the simulation trying to address like where I do the mosquito release, right? So suppose I have like an area. So half of the area has the very wet, so have a lot of water there, and the other half is very dry, just land. Okay, so am I going to release my Wolbachia mosquito on the dry region or on the wet region? So here is a setting where my wet region is like 2.5 times more wet than the dry. And initially, I just release a bunch of them. So the color indicates the infection rate. So I release on the left. Excuse me. And then I let the simulation run. I see that the infection established and quickly spread out into the wet region. So this is working well. Now I repeat the process, but release mosquitoes in the wet region. I see that, okay, it also spread into the dry region, right? And then to track the speed of this sweeping dynamic, okay, we examine the average infection level on the whole domain. So I see that when I release in the wet region, the infection established much faster compared to release in the dry region, where when the infection crossed the boundary from dry to wet, actually it slows down for quite a bit. So the idea here is like, okay, release both dry and wet is okay, right? But like releasing the dry region, the advantage is you need a smaller amount of infection to establish, but then it can slow down when it propagates across the jump. We further make the heterogeneity more substantial. So now my wet region is 5 times more wet than the dry. When I release in the dry, I see that my infection wave front stops at this interface. When I release in the wet, the infection still quickly sweep into the dry region, tracking the infection level in time. So we see that release in the dry, it never increase. It just it just stagnates at that wave at the interfaces. The reason because you see that is because when you release in the dry region, it established, you have a bunch of infected mosquitoes, it try to propagate into the wet region. But then in the wet region, there's a lot more natural mosquitoes that's already living there. So your wet, like infected cohort is not going to be like, is not going to compete over that natural abundance. It's overwhelmed by the amount of uninfected mosquitoes there. So it just stops there. Well, from the public health point of view, this can be good or this can be bad, right? It is good in the sense that your released mosquitoes are like contained in a certain region. So, if people feel concerned about this use of the mitigation, then, this is like a more contained strategy. The bad could be like if you do want your infections to spread out, right? Then you actually cannot do that but when you release in a dry region. Lastly, I want to consider the seasonality impact. So lots of the mosquito life traits, it's very sensitive to the variation in the temperature or the variation like in the rainfall or humidity in the region. So we parameterize a bunch of mosquito parameters, which I didn't show you here for the time constraint, but I want to show you the climate profile that I use here. So here is a case study based in Australia, where on the left, this is like different months of the year. So the temperature fluctuates between 29 and 20 degree in Celsius. And then this curve is the rainfall curve. So there's quite a long time of dry season. On the right plot, the solid curve here, excuse me, the dashed curve here, it is the variation in the abundance of the natural mosquitoes in the absence of Wolbachia release. So this is how the mosquito abundance vary in time. And then the solid curve here simulates the threshold of a Wolbachia release. So we're looking for like a smaller threshold for efficiency. So I see that the optimal threshold here is in September, where just before the most natural mosquito abundance takes off, or just before the rainy season starts in September here. I changed the climate profile to another country, Indonesia, where the temperature variation is more mild, right? So that's a temperature, and we still see a little bit temperature like seasonality in the rainfall. On the right, we still see that the optimal time for release by mosquitoes is around September, when before the natural mosquito population takes off. So we see this like very consistent trend in our seasonality study, which is kind of making sense because on the hindsight, the efficiency of my Wolbachia release depends on how many of the natural mosquitoes I need to compete, right? It's a competition between this infected and uninfected cohort. So if I can reduce the natural abundance, then it's easier for my infected mosquitoes to take over. That is like corresponding to this minimal here. And also this minimal happens before the rain season starts rather than after the rainy season's like over. This is because when you have the rain season starts, then this rain creates more breeding habitats for the mosquitoes. So that will be more efficient for these released Wolbachia mosquitoes to breed and take over the breeding sites. Okay, I don't think I have much time, so I'll quickly go just move on to the couple of like future directions that we can proceed with this project. So we can consider more spatial heterogeneity settings. So here I only show you like one dry patch, one wet patch. So you can consider more complex like configuration of the space dimension. We can also consider like repetitive release in time. So often the time in the field, people release like batches of mosquitoes, which is like depending on the capacity of the infrastructure to create these mosquitoes. For the modeling side, we can improve the model by doing the fractional diffusion on the spatial dynamics. And lastly, I haven't talked about the human side of the disease transmission. So it will be like an important feature to incorporate, like to predict the Wolbachia impact on actual disease outcome on the human side. So for this, I'm collaborating with Lauren Charles from Virginia Tech in the US, try to assess the potential impact on Wolbachia to control malaria, the Anopheles mosquitoes. So that will be it for my talk.
Thank you. Thank you so much for your wonderful talk. Very informative. Because I'm seeing we have only 5 minutes. I'll open the floor for any questions if you have. Anyone, please? Yeah, I'm seeing someone. Yeah, please go ahead. A raised hand.
Okay, yes, good morning. Thank you very much for that very interesting presentation, Dr. Chu. I am Novi Younger Coleman, statistician with the Caribbean Institute for Health Research. So I know we don't have a lot of time, so my quick questions are, is this body of work, has it been published or do you plan to publish? Because I'd love to be able to, sit and wade through it a little bit more, read through the work you've done, or if there's a document that you can share that compiles the results, that would be nice. And maybe another question I have is, which piece of software or pieces of software did you use to do the model development? Thank you.
Yes. So for your first question, the work has not been published yet. So I have like a previous bunch of publication on the related topic if you're interested in reading like a modified version of this model. And our plan is to get that submitted ASAP.
Yes, I'd be happy to do that. I'll probably
Yeah.
put my email address in the chat and maybe you can send it.
And then the second question is the software. So I use
Yes.
I code everything in MATLAB.
MATLAB. Okay, thank you very much.
Thank you. Anyone? Okay. While others are thinking, maybe I'm sure everybody should have a lot of questions. But one thing I'm wondering, how do we know that the mosquito is infected, which is with the bacteria, Wolbachia, right? How do we know? Yeah, I'm just,
You mean biologically, like how they examine it?
Yeah. I don't know, which is giving me, so.
Yeah, so I think what they do in the lab when they create this, the Wolbachia is not naturally found in the mosquitoes. Well, they can examine like whether they're infected by just like dissect the mosquitoes and then put them in a microscope and they can examine. Bacteria is quite big, so you can see whether it's in their gut, like in their stomach or in their tummy of the mosquitoes. So it's just by visually exam, and then, but like I guess like another interesting question is like how we introduce this infection to the mosquitoes. So they actually extract that bacteria from other insects and then transfer that through micro injection to mosquitoes they breed in the lab. And then they release those infected mosquitoes, like when the mosquitoes are still in the egg stage. And then they release the mosquitoes in the field. Yeah, and they do need to keep monitoring the infection level just by capture the mosquitoes like in the field and then take them back to the lab and examine like whether they can find any bacteria there.
Yeah, wonderful. So I'm sure, I don't know, I'm getting several ideas. Maybe this idea was is using in terms of in reality right now.
There's few trials that people have been implemented. So I think the Wolbachia strategy is very not very bad. It is like practical and mature for controlling dengue mosquitoes, like Aedes aegypti mosquitoes. But like I'm mentioning the malaria mosquitoes here, so that's actually still under development.
Okay.
Yeah.
Thank you. Maybe you have only 1 minute. But what is the R naught according to your study that you calculated?
R naught.
The value, something which your
So
reproductive number.
So the reproduction number for Wolbachia infection is 0.7. So it's less than 1.
Okay. Oh, surprising.
you like for COVID, some people estimate it can be 4 or 6, right? For influenza, it's like barely past 1. So you see like seasonal waves of influenza. So for Wolbachia, it's 0.7. That kind of explain like why you naturally don't see that infection in the mosquitoes.
So you need 3 at least to infect 2. Okay.
Yeah,
Oh,
exactly.
okay.
So
Okay.
that's why like you need to release a tons more, to get the initial infection like kick-started, and then it can spread later on.
Okay, very, very interesting, Dr. Chu. So we don't have a time, but I really wanted you come back to us again and give us some information even about your university so that our students can have an idea if you have any opportunities for our students, to do higher education. So I look forward, whenever you have a time. Thank you so much from the department. Thank you.
Yeah, feel free to share my email address with the participants or anybody who may be interested, and feel free to email me if you have questions.
Thank you so much for that. Thank you. Have a wonderful class. Thank you.
Thank you.
Bye.
Thank you. Very good presentation.
Hi everyone.
Bye-bye.
Hello Dipti.